usually gets not pushed to the side in terms of career skills, but it kind of does. Because a lot of students are just trying to get their generals, but students in the sciences in their first 2 years really need to get experience with research and research is on the job preparation for students in science. So some of this started with the CCC mini grant program, where I was awarded a grant to continue doing my bluebird research that I did for my PhD with students. And that led to some really interesting opportunities in addition to Project GPS, where we've collaborated with the Materials Center, the Nanoscience Center, Materials and Nanoscience Center in Lincoln at UNL, because this here is a photo of a feather under the electron microscope. But we're getting students hands-on animals, getting them hands-on experience. We've got a couple of different projects going on. So we're working with the bluebirds still. We have a smart nest box project where we're looking at integrating technology into nest boxes. We have a project going on with birds and technology where we're looking at these swallows that we have found, where I have another mini grant as well that deals with the different plumage colors, the feather colors in these swallows that we found that aren't normal. These white patches aren't supposed to be there and are similar to a cliff swallow, but we're not really sure what's going on. And so we've got a research project going on where we're investigating that in concert with the Nanoscience Center and utilizing their technology and equipment. So in our first year of our cohort, we gave out about $70,000 in scholarships. And at this point, I think we're up to about 120,000 or so that we've given out to support students. So it's really exciting. Our relationship with the Nanoscience Center started out as a tour, and we figured out that we had some really common interests in terms of research goals, and especially with Katie's fungal material. Getting into materials testing is something that we're going to start this year. So we're really excited to start those visits up again soon. We presented at conferences. We presented at the Nebraska Academy of Sciences. In addition to that, not last summer, but the summer before, we took some students to a conference in Alaska. Where we presented our bird research at the National Ornithology Conference. This summer, we attended the National Congress for Conservation Biology, or North American Congress for Conservation Biology virtually, where we normally would have traveled to Colorado, because we try and get the students out at least once a summer to have some type of experience. So these are just some of the activities that we do with our students and with the scholars to get them experience, to expose them to different opportunities in the community. Like this, we have them usually go on tours of industry. We're not able to do that right now, so we're actually setting up a speaker series. So now we reach Katie. So Katie was a student at CCC before she became part of Project GPS. And she grew an 8-foot canoe out of mycelium. Pretty incredible. And so we were just absolutely ecstatic when she found out about our program and wanted to join. As a result of that, we have been collaborating with the Nanoscience Center looking at her material, and we've found some really cool stuff. These photos are photos with an electron microscope. And this one on the left is at around 3,000x. And this one on the right is at 89,000. And so we found that it actually acts as a semi-permeable water sealant. The skin of the fungus does. It's very, very cool. So what we're doing, and what Katie's going to talk about in some more detail in a moment, is we're using the mycelium to try and help bees. Because there's some research that Katie will talk about in a little more detail that shows that fungi and bees have relationships that can help benefit the bees and the bees' health specifically. We're dealing with a pollinator crisis across the country right now. So we're really looking to try and help augment that locally, regionally, and then also nationally. We're about to submit this project to the MIT Student Lemelson Innovation Competition. So we're really excited about this project and what we can do with it. So with that being said, that's a little bit of a background about who we are, how we do what we do here, and how we're able to do this project. So next, I will grab this here. And Katie will tell you a bit about fungi.
So when you think of fungi, what do you think of? If you're like most people, you're probably going to think of mushrooms, but it's so much more than that. So mushrooms are just a fruiting body, meaning that this is just a reproductive structure. The actual body of the fungus is known as mycelium, which is exactly what we've grown these bee hotels out of. The mycelium grows through organic material. In this case, we used ground up pellets to make it more sustainable. And it grows through it and then forms the skin around it, which, like Dr. G said, is non-porous and semi-permeable. The mushroom needs air to live, so it's not going to let enough water in to drown it, which is why it's so good for boat. Whenever we took it out, we floated for about an hour, and the boat only gained 2 ounces of water weight. So when these bee hotels are exposed to rain, they shouldn't absorb hardly any moisture at all, just enough to do another round of fruiting or growing mushrooms. Fungi grow on organic material, and basically their job is to actually break it down into smaller, more useful molecules and atoms for plants, microorganisms, macroinvertebrates. So they really make healthy soils. They're the reason we have such great prairies here in Nebraska. And without mushrooms or fungi in general, imagine if your trees that fell never broke down, or if plant material never broke down and composted. We would be up to our necks in dead organic material. Don't even get me started about animals. So mycelium has a lot of applications. One of my favorites is bioremediation and water and air filtration. There have been multiple studies that show that different types of fungi can break down different harmful pollutants. Like oyster mushrooms have been shown to break down oil. A specific type of oyster mushroom, king oysters, have been shown to break down Agent Orange. And shiitakes, heavy metal. So almost anything you can think of, fungi will break it down. Building materials, obviously, we've grown a boat and bee hotels at this point. I've also grown furniture like tables and chairs. We grew some flower pots, multiple items, using whatever we could find. It's already being used as a styrofoam replacement by companies like Ecovative, who supplies packaging for Dell and IKEA. One of the main reasons that it's great for packaging is it's a single-use item and most people just throw it away. So instead of throwing away an item that's going to take thousands of years to break down, we're throwing away something that's going to help other things break down. And that is a beautiful thing. It's already used in antibiotics like penicillin, and it can be used as a fertilizer. Since it is breaking down those organic compounds, what better thing to feed your plants than organic materials? And then mycopesticides is another fun one. Paul Stamets is a well-known mycologist. He does a lot of studies with fungi, and he actually came up with a way to feed ants a certain type of fungi, and then they take it to their queen and it kills the entire colony. And that scent that is put off actually keeps other ant colonies from moving in. So anything you can think of, fungi can probably do it. The other great thing about fungi that we found is that they actually help bees. So Stamets, who I mentioned, teamed up with Washington State University's Steve Shepard, and they got together and started feeding honeybees a mixture of the exudates, which is just digestive juices, like our stomach acid, and sugar water. Then they put these into little bee feeders and they fed it to some colonies, and then they also had their control colonies. And they tried a few varieties of Ganoderma, which are these shelf mushrooms that we're using as well. And one species in particular showed really drastic results in decreasing certain viruses that have been known to cause colony collapse disorder. So if you know what colony collapse disorder, honeybees are basically disappearing. Whole colonies are disappearing and nobody can figure out why. And there's 3 things that a lot of people think could do it. And the deformed wing virus and Lake Sinai virus are one of those things. Whenever they did feed the bees the exudates, the testing colonies actually showed like 49-fold reduction in virus and 49,000-fold reduction in Lake Sinai virus. And there have been other studies that show that these viruses do cross over into our native pollinators. Will you go to the next slide?
Yes, I can.
And our native pollinators happen to be solitary bees and wasps. Now, most people don't think much about solitary bees and wasps because we're human and we're in it for the honey. But pollination is really important. So there's 4,000 native species of bees and wasps, like solitary bees and wasps, in the United States. 4,000. That is so many. And a lot of times people are afraid of them because you think of bees and wasps, you think you're going to get stung. But these are gentle bees. They don't have a hive or a colony to protect. So as long as you're not directly threatening them, you actually probably won't get stung. Maybe if you accidentally step on one. But the females actually go and they'll create their own nest. Each female has its own nest. And generally it's in a cavity, like in a tree or maybe in the ground, old stems of plants. That's why it's a good reason to not cut your garden down until the spring. You might be taking away some of your pollinators. Next slide. So the life cycle of these bees, when they go into the cavity, they'll make brood cells. And at the very back, usually there's about 6 to 8 brood cells in each of these cavities. And at the back, you'll have your female larvae. I don't know how they can tell, but they do. So they lay the females at the very back so that they're the most protected. And then all the male brood cells go in the front. And then they pack enough pollen with little leaves that they use to make the cell. And the pollen feeds it throughout its pupal phase. So it goes from egg to larvae to pupa and then adult. And then in the spring or summer, those will emerge and there'll be full-grown bees and wasps and keep going with pollination. One of the reasons why these solitary bees are so good at pollination is because they don't have pollen sacs. So like normally on the little knees of the honeybees and bumblebees, they have pollen sacs, and that's where they carry all the pollen back to their colony. But the solitary bees and wasps, they do more of a belly flop. They just kind of slam into the plant, so it's all over their body. So that means that the chances of transferring that pollen to another plant are drastically increased. Honeybees are estimated to have a 5% pollination rate, whereas solitary bees and wasps have more of a 95% pollination rate. That's a big deal. So on the right, obviously we have another picture from the scanning electron microscope, and then on left, right. And then right. When we looked at it, it was really intriguing because you couldn't see any pores, which clearly shows why it would be a good water sealant. We did not have to treat the canoe in any way to make it buoyant and not retain that water. And then after each time we took it out, it actually grew mushrooms, which is kind of what we want to happen with our bee hotels, since the exudates or digestive juices are what could benefit the bees' health. And to build these bad boys, we went really high tech, and we used some cardboard boxes covered in plastic sheeting. Here is one that we haven't filled yet. As you notice, we have the inside of this high-tech cardboard box covered in plastic sheeting, and then the outside of the inner box, and that's what's actually creating the inside portion, the hole. Eventually, we'd like to get to the point where we're making the nesting block or the cavity insert from mycelium as well. but we'll actually be baking those to make the mycelium die or become inert. And that way we can prevent it from growing in and closing those holes, or potentially growing into some of the larvae and trying to consume it, because that would defeat the purpose. So next I'll show you a video of the unboxing of our bee hotels. This is the very first round that we made. And this was right after the beginning of the pandemic, so I had to do it by myself. I had my camera actually sitting on top of the humidity chamber, and then I just play and started going. It was a little bit more difficult to get the inside box out, but eventually I found a way that it came out pretty well. And voila, the outer shell of a bee hotel. The first model we actually put up in the pollinator garden on the Columbus campus in July, and within 2 weeks, oh, I don't give that away. We put it up in the garden and we started to wait. We had a little bit of an issue with mounting, so we ended up having to make this sort of cage around it, but future models, we're going to try and adjust our mounting technique so that it won't be as boxy and bulky and maybe look a little bit sleeker and streamlined. So in 2 weeks, guess what happened? Huzzah! We had a wasp, actually more than one. At this point, a lot of the holes have been filled in. So this is really exciting because clearly that shows that this does increase habitat because they're using it. So we've effectively increased their habitat for nesting. Now we need to figure out how we can quantify if this is affecting their health. You might think, you can't do a doctor checkup on a bee. What are you going to do? They don't live all together. They don't have colonies. How are you going to catch all these and find out what they are?
We've got some plans. So what we're going to do is, and what you normally do with a bee hotel is you leave it out all year long. So this prototype, we're going to find out a lot from this. One, we're going to find out how it weathers all through the year, through snow and the winter weather. And then what we need to figure out is a system of basically putting we're going to end up putting a plastic bag around the front of the hotel, so around the spot where all the little holes are. Actually, I have a better photo I can show you of the occupied holes there. Hang on just a moment here. Where's our...
We've made quite a few PowerPoints at this point.
Yes. We got a couple of different irons in the fire here. So this is our PowerPoint for the MIT invention competition. But this picture here shows really, really nicely that we have, you can see all these little grass, these areas where there's little bits of grass in those tubes. That means that someone's nesting in there. And so there are a couple of different options that we can use to figure out how they're nesting and if we're affecting their health. One problem that we're going to have, and one reason why we need to contain who's in there, is because doing background research on this project, okay, so doing scientific research, you have to find out what other people have done before to base what you're doing next. And so I started doing a deep dive into looking for solitary bee and wasp information and specific species. And there's very little information. We may end up identifying species of bees in Nebraska that have never been identified in Nebraska before, very likely, because of the fact that solitary bees and wasps, there's so many of them, they are incredibly understudied. So we are poised in a very unique and an interesting and exciting place to be able to learn a lot and contribute to multiple fields of science in terms of gaining new discovery science, new information in terms of, one, providing a renewable, sustainable way to increase habitat for pollinators generally is a big deal. let alone the fact that we can discover more about these species themselves, identify species, and then potentially look at and gauge the health of these species. So we may be able to, once we identify which species are using these boxes and their larvae, we may be able to pick out or work with other bee experts on looking at the health of the larva in some way and taking some measures in some way, shape, or form. We're still working this out, which is part of the exciting part. So as Katie said, for example, the insides right now, these are cardboard recyclable straws, these little tubes. And what we're going to be doing next week actually is we found CCC used to actually make their own pipettes. And so we found bunches of glass tubing that we are going to cut to size to put in these hotels back here. So if you can see this area here, this is our growth chamber. And we currently have 1, 2, 3, 4, 5. We currently have 5 new hotels growing. And next week, we're going to be measuring the depth of each of the hotels because they're all different sizes and depths. And then we're going to cut these glass tubes to size so that they'll fit into these boxes. And then we're going to create these inner boxes, the inserts, and then insert the glass tubes into the inner box to create those tubes for the bees to nest in. So we're really, really excited. We've got actually a really, like, one of these is really big. I don't know if you're really
and it's growing mushrooms.
See it, and it's growing mushrooms. So it's really, really exciting in terms of what we're doing here and what we're able to do. And then, we haven't even touched on looking at the exudates coming out of the mushroom, where we may be able to actually collect some insects, collect and extract some of these exudates, and then maybe do some experiments testing these exudates on some of these insects and seeing what happens. The interesting thing about insects is you don't actually need special permissions to do experiments with them. So we actually, the field is open for us to kind of play around and really experiment and let students come up with ideas of how to explore the scientific method, but also contribute to different fields of science in a really meaningful and lasting way.
And now, so I said that we're using a species of Ganoderma, but I want to make clear how many Ganodermas there are just in Nebraska. So we're using 2 main kinds. We have our red. This is a little bit small of the mushroom, but you can tell it's really red. And then the white. And we're using both of these. This one is grown from a lab culture. So it's used to growing indoors. It's normal for it. This one is a Nebraska native that we actually just found on a tree stump. So we brought it back to the lab. We cultured it, transferred it, and now it's become these bee hotels.
And that's what this is, right?
This one is the red.
Oh, this one is the red. Okay.
For some reason, when the red grows mycelium, it's more white. And then the white almost looks red.
But isn't this time around it different?
Yeah, this time it happened exactly opposite.
Yep. And the new ones, the white is white and the red is red.
So there's a lot of unknowns in the field of fungi, which is why it's so exciting.
It is. It is.
And then there's also the artist conch. So you probably are used to seeing these. These are the most common ones around Nebraska. People actually used to pick these, and then on the bottom side, you can carve into them and make your own artwork, and it looks really, really cool. And here's the biggest one I've found here in Columbus. This is a good-sized mushroom. They're actually really woody, and you can tell they're annual. They grow each year, and you can tell how old it is by counting the layers of pores that it has. It's kind of like a tree. And it grows off a tree. Let's see, this one's 1, 2, 3, 4, 5 years old. And I pulled it off the tree, and now it won't reach 6. Sorry. They also create these spores. You can tell kind of this looks a little dirty. It's actually spores on top of there. And the spores can be beneficial for your health as well. Oh, I got it all over my pants. The spores are the reproductive part, so they'd be like the sperm and the egg. But for spores, we label them as positive and negative because how are you going to tell?
Fungi don't really have sexes.
Yeah, they're just are. And I propose that we change flora and fauna to the 3 F's of flora, fauna, and fungi because they're often overlooked, just like our solitary bee friends. So we're combining 2 underrated, underacknowledged areas.
Understudied areas as well.
And really trying to lift those up and bring it to the attention to as many people as we possibly can. Yeah. I will literally talk to a stranger on the street if they will let me.
And we're really looking to make things happen professionally. Katie here with this. We're looking into trademarking the Mycohotels next week. And then eventually there'll be a patent for Katie on the process of actually creating these hotels and coming up with them. So it's some really exciting stuff. We are thrilled to be doing this. Our goal is to make 40 boxes by the end of the year. We've got 5 now. We did just come up with a little bit of a hiccup where we now actually have to make our own mushroom spawn, which is the mushroom itself combined with sawdust in a bag, right?
Yes, basically.
Essentially.
Mycelium.
The mycelium, the mushroom, the mycelium, the body of the mycelium itself, which the body of the mycelium are those hyphae, or they are hyphae, called hyphae, but those string-like elements. So we have to make that. We used to be purchasing that, and now we are going to be actually making that ourselves. So it's actually really kind of cool. We are doing absolutely every single step of this project, of the production process, in-house here at CCC. We will be utilizing probably UNL for some autoclaving, for some sterilizing. But for the most part, these will be produced right here at CCC Columbus, and it's really cool and it's really exciting.
And then in the future, we thought this out a long ways. If this will work.
Do you want to go back?
Yes, the large scale production flag. So we've actually considered how this could be implemented on a large scale already. So farmers would be able to put a large version of a Mycohotel on their property. We would mount it onto a trailer, and you could actually drive these in next to fields. And then this would increase your pollinator habitat right next to the crops, which in turn should increase the yields because more pollination means more plants and more fruit or corn or soybeans, whatever they're growing. And then after it's been used, eventually it's going to break down. That's what fungi do. They break everything down. So instead of just throwing it away and being a waste of wood or whatever resource you're using, we could literally till them into the ground and use them as a soil amendment. It would feed the plants. It would feed macroinvertebrates. Everyone's winning here.
So it's a really great eco-friendly use of recyclable materials, sustainable way to help increase crop production while at the same time helping pollinators, which is usually not something you could do at the same time. So we're pretty excited about the possibilities going on here.
And for commercial production, the only things that we would really need are extra large molds, which we've decided we could probably make using the injection molding. It would also need filters because fungi breathe. They don't actually breathe, but they use oxygen. They need access to oxygen. So you would put some filters on there, and then you'd have to make giant batches of spawn, like bats. And then you would want to have this grow in optimal mushroom growing conditions, so 80 to 100% humidity with about room temperature, maybe up to 85, 100 degrees would be tops.
We've created our own humidity chamber here by taking plastic sheeting and wrapping it around shelving. But we are going to be purchasing a little indoor kind of greenhouse setup as well, so that we can get multiple rounds of boxes going at the same time.
And I'm the humidifier right now.
Until that comes in.
So once we get the humidifier, I'll lose one job. But I'll enjoy it while it lasts.
All right. Well, that's what we have for you folks.
Any questions?
Questions, discussion, thoughts? I can unshare. Do you want me to unshare, Ben?
You could leave it on if you
Okay.
want. We don't, or you can feel free to enter any questions in the, hold on, Megan, in the chat box. Um, and then I'll ask the question since everybody's muted. But we do have a question. Um, can fungi, or are you getting feedback from me?
We can hear you. Yep.
Oh, okay, all right. In fungi, so a major problem that we did here this morning when we went on the Hastings Solar Farm tour, they have a well field out there that supplies water to the city of Hastings. And a major issue that they have in central Nebraska is that there's a lot of uranium that filters through the cornfields due to irrigation, and that uranium just keeps increasing year after year. It's getting higher and higher every year. I did see your slide about filtering out heavy metals. Would mushrooms be able to filter out uranium potentially?
Yes.
Oh,
So it's actually, oh, now I'm getting feedback. It's actually really cool because they've been using mycoremediation, which is remediation using fungi. They actually brought it to Fukushima. So I don't know if you guys remember, Fukushima had a disaster basically, and there's all this nuclear waste going into the ocean and everywhere, and they brought fungi to help clean up that mess. Also at Chernobyl, one of the only things that's growing inside of that reactor is a fungus. So they love the extreme. They love the dirty, the nasty, the stuff that we want to get rid of. Fungi are our best friends for doing that because they already know how to do it. We'd have to come up with some new system. They do it naturally. It's their job. And that's actually one of the other big ideas. I have a lot of big ideas when it comes to fungi. So one of the main ones that I really would like to see implemented in Nebraska is utilizing fungi around in the ditches around every single crop and livestock enclosure. And by doing that, you could specify the type of fungus to what is running off of there and actually improve our groundwater, reduce the pollution, reduce all these things being moved to places we don't want.
Which are the mushrooms that digest the heavy metals best?
Heavy metals, shiitakes, some species of oyster mushrooms. There's also mushrooms that love to eat coliforms or E. coli. So you spread that around near your feedlots. And there's one for everything, I swear. If it hasn't been found yet, we just haven't looked hard enough. It's out there.
Time.
Yeah.
Well, that's great news because I heard from Hastings Utilities today that they have put in like a $13 million system to filter out like uranium, nitrates, and they just keep having to increase that filtration system. So, that might be something to look into. I know you're already doing great work on with many different applications, but that could be another avenue, that this could go into.
That's actually my concept for my future thesis statement. Bioremediation. But instead of having a $13 million system, we literally add wood chips.
Right.
So this is a much healthier, environmentally friendly, sustainable,
Cost efficient.
It checks all of our boxes, really.
Yeah, definitely. Does anybody else have questions? Because I can keep asking questions. But we don't have any questions yet in the chat box. But I did have another question. So are you making different variations of your boxes? So I know like with this one, it was your prototype, but you mentioned possibly making up to 40 different boxes. Are they all going to be a little bit different based on what you learned from your first prototype?
A little bit. We want to change the mounting strategy And then obviously they'll be grown out of 2 specific species of fungi. So we'll probably have 20 of the red variety and 20 of the white variety. And then we want to have the inner nesting block actually be made of mycelium and a way to mount it without the bulky wire frame.
So there may be, for example, on our next batch, we kind of forgot to do it with this batch, but we may grow like chicken wire into the back so that we can, when we go to mount it, we can easily kind of the mounting system is actually grown into the fungus itself, into the product itself, because the cage that we put around this first prototype was so that we could attach the pole, and it's just a garden stake that we're putting it on. So we're going to look at experimenting with putting either hardware mesh or chicken wire on the backs of these and letting them grow in.
I'm a proponent for wooded lattice because the fungus will actually eventually eat it. And chicken wire, we would have to figure out how to get it out before we use it as a fertilizer or soil amendment.
That's true. So we've got some things to experiment with in terms of figuring out the best prototype and the best model that's the sturdiest, that still does the same job.
I'm going to grab the prototype that didn't work.
Oh, yeah, go ahead. The other thing that we're starting to do is looking more at the production process itself. So we'll be taking measures of like these boxes. These boxes are just from my basement. And so they're all of different sizes, different shapes. And so we're starting to take measurements of the inner box, the outer box, the depths, the weight that we end up with at the end after they grow. And to be able to track the speed of growth and the rate of growth based on the size of the mold. Because what we'll eventually look to do is 3D print molds of specific sizes for specific needs. So if you have for your own personal home garden, this size is probably fine. If you're wanting to do something a bit larger, okay, you could have an option like this for a much larger garden if you're growing some more crops on your home property. And then you start to think about the larger scale molds that you would need to go commercial, which would be, very, very large, the size of a small shed even, maybe. So these are some of the things that we're looking at doing. We do have access to 3D printing here at the college, which is really exciting. So what we're probably will aim to do is to get once we get the kind of the growth rate of certain sizes, we'll aim to print 3D models that we can reuse of these molds so that we no longer have to use the cardboard boxes, but so that we can use these molds that are reusable. And that we can, again, style in different shapes, different sizes. You can make them into pretty shapes. We have another example here of looking at trying to use concrete and also trying a different shape. I'll let Katie describe this a little bit.
So this is rags that have been dipped in concrete and then molded to shape. We're kind of going for a flower. And then this is another rag that's inside of it. And the substrate or mushroom spawn is all around the edges. But you'll notice it didn't grow the best in the concrete. I don't know if it didn't have enough air. It did grow mushrooms, but it didn't skin up like all the other materials did. It didn't grow this nice white. But it doesn't fall apart. I mean, it's one thing. It just doesn't look quite as good as the other ones.
Yeah. So we're experimenting with some different types of materials to use as well. So we've got a lot to focus on in terms of trying to think about how to make this more sustainable, and how to kind of ramp it up, because we have requests from all over the state for these bee hotels on different gardens, different farms, different properties. It's really exciting. People don't even know if it works yet, and they're like, "We want one." And so now we're really excited to at least be able to say we know that they will nest in this structure, and so we're pretty confident to say that at the very least we will be providing a natural increase in habitat for our natural pollinators, and then the bonus is, does the, living amongst and in this mycelium, does that actually help the bees and wasps themselves become more prolific? So we've got our work cut out for us, for sure.
It's also a really great insulator. So if we were to have a particularly harsh winter, the larvae that's inside of those should be just fine. It provides more insulation actually than just wood.
Right. So, I guess I'm still not questions.
I'll ask a question. Hey, chat, if you were to grow something out of mycelium, what would it be?
Oh, yeah. We'll see if you want to grow something. I know David said he wanted to grow an umbrella.
An umbrella would be kind
Ooh,
of interesting.
that would be interesting.
As long as the wind didn't come up. But hey, you could probably use it as a boat too. So
Yeah. Serves many purposes.
Yes.
So after you test all these different prototypes, and so many people are wanting, the Mycohotels, when would you feel comfortable like giving them out to, or if people are selling them, after you do, a year's worth of research, or are you like willing to give them out like now, if people on this webinar are interested, what's that process look like, I guess?
Well, personally, I want to include the community in the data collection process. I won't really feel comfortable selling it as a commercial product for like 5 or 10 years. You really need a lot of data and make sure that you're not like harming them somehow, which you wouldn't even know without years worth of data. So next year will be the first year that it's open. You won't have to pay for it. You can just request that you can house one on your property, and then all that you would have to do is give permission for a research student, one of the GPS cohort, to come to your property maybe twice a month, every couple of weeks, to check on the progress of the bee hotel and to monitor any populations or changes. So the more places that we can put them, the better data we'll have.
Yeah.
We have places that are completely surrounded by organic cropland, places in towns, places out of town where they use maybe glyphosate and other pesticides. As many places as we can get.
Yeah, because we're just starting out, and we know, like Katie said, at this point we know that they'll live in them, and so at the very least we can augment housing for these populations. And as she said, until you test something and have some really solid data where we can really confidently say, not only does this increase the habitat for these organisms, but it also helps their health, we need some more data to be able to make some of those statements. But we do feel good about giving these out to folks who want them. So anyone that would like to put it up in their garden, for example, please feel free to request one from us. We can ship them. We'll figure it out. But we definitely are going to be, it's going to be volunteer this summer, and we'll be deploying them out this spring as soon as it's time. So we're going to be continuing to grow them all year long and basically grow as many as we can so that we have as many as possible to give out, because the interest that we have in this project is overwhelming.
Oh yeah, I definitely want one in my community gardens.
Yes, and we can make different sizes for different sized gardens as well. So if folks give us an idea of, hey, I have a, this square footage garden, we can look at scale as well, to make a larger hotel for that specific property.
Yeah.
And you did mention the patent process, and that you were so are you patenting the process but not actually the hotel? Is that correct?
So what we'll do is we'll trademark the hotel, which will give us the Mycohotel name and I believe the concept, and then we'll patent the process of the mold and the growing and the the process for that idea of the bee hotel, of using it for a bee hotel. Because in patenting, you have to be, and trademarks, you have to be really careful about the types of concepts and ideas that you're talking about, making sure that maybe someone has a similar process, but it's a different application. So that's where we're at, where there are other companies that do similar processes, but our process is still a little bit different, and we have a completely different application and usage of the end product.
I like Mycohotels, their process, they actually use corn stalks and hulls. So they get the big bundles that here in Nebraska would go to the ethanol plant, and they grind those up and then inoculate it with their fungi. And then they use injected molds, and they'll have a tray that has 8 or 9 different packages on there, fill it up, and then they'll put it into a special grow room, and then they wheel the whole cart over to their drying room. So they've really got it down to a science, but they're using a different material to grow it into, probably a different species. Since ours are native to Nebraska, that's really cool. Well, except the red one.
But we're really excited about the one that's native to Nebraska too, because we really think that the native species are going to be more helpful to other native species. And so when we picture this going national, we would actually want to go and actually collect and cultivate species, because you've identified this as Ganoderma megaloma, which there aren't many accounts of it, if I remember correctly.
Megaloma is not found very rare. Lucidum, Ampellantinum. There's a lot.
So it's a rare, almost a more rare species that we're working with here in Nebraska for these hotels, which is kind of exciting. But a lot of species of fungi also haven't been documented. So that's the other cool piece about when we go national with this, is going out and doing some documenting of different fungi in the area and finding those species.
There's one I found only at Indian State Caves, which is right near the Missouri River. So that one would probably be more specific to that area. But it was so beautiful because it had purple too. And it's called Ganoderma curtsii, and every single species I find, I get more excited. I don't know if it'll ever stop. I think it's just going to go forever.
Yeah, I mean, maybe when you do the science building, you'll have your own lab there now.
Yep, in 2 years. Maybe I'll come back for it.
Yeah, well, we're hoping that she'll come back and visit us. But when Katie transfers, we've talked a lot about her transfer and what she's going to do, and she'll continue her research wherever she goes to transfer, and we will continue collaborating on this project going forward. So when she leaves CCC, she doesn't leave us or this project in reality. We'll always be connected through this, and we'll always be collaborating and working together.
Oh, that's great to hear.
It's really exciting.
Yeah, well, I don't want to take all your time on a Friday afternoon, but I do want to thank you for sharing this with everybody. We would like to have, this in person at some point. Maybe next year, Katie will come back from wherever she's at, Pollinator Fest in the fall, and be our keynote speaker for no, so we typically in the past, when we held the event in Grand Island, we would get about 300 to 400 attendees.
Wow.
So it was growing, and with this year we had to do more virtual. But we hope to have it, next fall again in person. And it was growing every year, and we're just taking a little break this year.
Yeah. A necessary one as well.
Yeah.
Unavoidable.